Hello everyone, welcome to the new session of cell cycle and cell division. This is the third part and we are going to discuss about meiosis, the type of division that is called as meiosis. Okay, so the production of offspring by the sexual reproduction includes the fusion of two gametes, right? So there is a fusion of gametes. One is male gamete and another one is the female gamete. So if we take a gamete, suppose this is gamete N coming from the female individual, right? That means it is an egg. Okay. And we are having a sperm that is coming from the male individual. So this is the sperm and this is also haploid and means haploid set of chromosomes. Okay, so with a complete haploid set of chromosome, here it is showing the production of offspring. So new offsprings are produced by the process of sexual reproduction, in which the fusion of two gametes takes place. Two gametes unite and form a new individual, right, by the process of sexual reproduction. So the gametes are formed from the specialized diploid cells. So these gametes here, the egg and the sperms, are formed from the specialized diploid cells. They are special cells, okay, which are present in the male and female reproductive organs. Okay, so this specialized kind of cell division that reduces the chromosome number by half results in the production of half daughter cells. So this specialized cells is actually what? Diploid. It is having 2n, means two set of chromosomes, right? But in their gametes, when it forms a gametes, right, whether it is a egg, that is a female gamete, or a sperm, that is a male gamete, they produce haploid cells. They produce haploid cells and those are called as gametes, right? So they undergo a special type of cell division, that reduces the chromosome number by half. So if the diploid cell is having 46 chromosome, right, means 23 pair, as in the case of human beings, then it will form 23 chromosome. That means single pair, that is 23 chromosomes. Okay, this kind of cell division is called as meiosis, but it is not very simple. We have discussed about the mitosis, right? It is not as simple as mitosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms. So this is a type of cell division that ensures that it must undergo the haploid phase and form gamete, right? So in the life cycle, sexually reproducing organism, right, undergoes this haploid phase, whereas the fertilization restores the diploid phase. And after the formation of haploid gametes, they fertilize. So here again we can see egg and sperm. So this is egg, this is sperm, haploid cells, both are haploid, right? And when they fertilize, they form 2n, that is the zygote, and again it is having 2n, that is the diploid phase. Zygote is showing the diploid phase, right? So fertilization restores the diploid phase. Okay, so we come across meiosis during the gametogenesis. So the gametogenesis is a very important process in the cell division, right? And this occurred due to the process of meiosis, the type of division that is meiosis. Okay, and it occurs in both the plants and animals. Meiosis leads to the formation of haploid gametes. Yes, We have just now discussed that for the forming of the haploid gametes, the special cell must undergo this type of division that is called as meiosis. Okay, so the key features of meiosis are as follows. Meiosis involves two sequential cycle. We have seen mitosis, that is having one sequential cycle. Here we will have a two sequential cycle of the nuclear and cell division called meiosis 1 and meiosis 2, right? But only single cycle of DNA replication occur. There is no that we know that for the doubling of the chromosome, there is a process of DNA replication that occur in the S phase of the interphase, right? Okay, so the DNA replication doesn't occur two times, as we can see that two sequential cycles take place, but there is a single cycle for the DNA replication. Okay, so let us see how does it happen. Okay, so meiosis 1 is the initiated after the parental chromosome have replicated to produce identical sister chromatid at the S phase. So again in the S phase, synthetic phase of the interphase, DNA replication takes place, right? So the parental chromosome have replicated in this phase and produces the identical sister chromatids. Now they are having sister chromatids. Okay, so meiosis involves pairing of the homologous chromosomes and recombinations between the non-sister chromatids. Very important process. Okay, so the homologous chromosomes comes and pair like this. This is one chromosome and this is another chromosome. Suppose this is a chromosome number 1. So it is having pair, right? Now for chromosome number 2, we will have like this pairing. For 3 in the same way, right? So there will be a recombination between these two, okay, between the non-sister chromatids of the homologous chromosomes. Okay, so we will see in the coming slide that how does it happens. 4 haploid cells are formed at the end of meiosis. So what will be the result of the meiosis? Okay, so the 4 haploid cell will form at the end of meiosis. That will be meiosis 2, after meiosis 2. But we have seen that in the mitosis, at the end of mitosis, we are having just 2 cells, right? But here 4 cells will form, and the difference is that the cell will having half the chromosome number. That is the haploid set. Okay, so meiotic event can be grouped under the following phases. We will have 2 phases of meiosis, meiosis 1 and meiosis 2. In the meiosis 1, we will have prophase 1, metaphase 1, anaphase 1 and telophase 1. In the same way, meiosis 2 will have prophase 2, metaphase 2, anaphase 2 and telophase 2. Okay, so we will see how the cell divide under meiosis 1 and meiosis 2. So meiosis 1, okay, so the first division is the prophase 1. The prophase 1 for the first meiotic division is typically longer and more complex when compared to the prophase of mitosis. So if we compare to the prophase of the mitosis and meiosis, there is a drastic difference in both the type of mitosis. Okay, both the type of prophase. So it has been further subdivided into the following 5 phases based on the chromosomal behavior: lactotine, zygotine, pacotine, diplotin and the dikinases. So we are having this type of division, okay, this type of subdivision in the prophase 1. These are all the subdivision phases of the prophase 1. So first one, during the lactotine stage, the chromosome become gradually visible under the light microscope. So they become condensed and they can be visible under the light microscope. The compaction of the chromosome continues throughout the light lactotine. So the chromosome become more compact, okay, condensed, and they can be seen with the help of light microscope. So this is the lactotine stage. We can see in the figure, right? The chromosomes are quite visible under the light microscope and the chromosomes become compact. So this, it is followed by the second stage of the prophase 1, that is called as the zygotine. So what happened in the zygotine? During this stage, the chromosomes start pairing to, and this process is called the association, and the synapse can be seen later on. So what happened? The chromosomes come and pair. So this is chromosomes and they are in the paired form. Because in the replication process, every chromosome has made a copy of it. So this is the copied one, at the S phase. So they become paired, like chromosome number paired, chromosome number 2 paired, chromosome number 3 paired, and so on. So every chromosome get paired, right, at the zygotine stage of the prophase 1. Such paired chromosomes are called as homologous chromosome. Now they are homologous chromosome because both the chromosomes are of same type. The chromosome number 1 will pair with the chromosome number 1. Okay, the chromosome number 1 will not pair with the chromosome number 2, right? So this pairing is the homologous between the homologous chromosome. Electron microgroove of this stage indicate that chromosome synapse is accompanied by the formation of complex structure called the synaptonemal complex. So in this stage, what happened? Each chromosome now opens and the chromatid can be seen like this. This is one chromosome and this is the paired one, that is the homologous chromosomes, and it is showing its chromatids. So this is the chromatids, right? Chromatids. This is the homologous chromosomes, right? So we can see in this way. Later what happened? This is one chromatid and this is another chromatid. This is one chromatid of one chromosome and this is another chromatid of another chromosome of same type. Now they interchange their genetic material by forming a complex structure which is called a synaptonemal complex. And at the point where these two arms or the chromatids join, that is called as synapse. We will later see how it happened. So the complex found by the pair of synapsed homologous chromosome is called as bivalent or the tetrad. So at this particular stage, it is called as bivalent. This is also called as tetrad or the bivalent. Now I'll tell you why it is called as bivalent or the tetrad. So here we can see two chromosomes, right? Two chromosomes. So they are called as bivalent and they are the sister chromatid. This is one chromosome shown with the red color, that is one chromatid, and this is the second chromatid. One chromatid and this is the second chromatid. Again, for the blue chromosome of the same type, this is the one chromatid and this is the second chromatid. Okay, so this is the bivalent structure. And why it is called as tetrad? So if we consider this, and this, four chromatids are there, so it is showing a tetrad structure. Right? So bivalent or a tetrad. However, these are more clearly visible at the next stage. next stage. So the first two stages of prophase 1 are relatively short-lived as compared to the next stage, that is called as the packetine. So at the packetine, the tetravalent stage can easily be seen. So during this stage, the four chromatid of each bivalent chromosome become distinct and clearly appears as a tetra stage. So at which stage the tetra stage clearly can be seen? So that is the packetine stage, right? So in the figure here also we can see the chromatids of blue chromosome is light blue and dark blue. In the same way, the chromatid here of the second chromosome is shown with the red and yellow. Okay, so it's showing the tetrad stage. Now, this stage is also characterized. This stage is also characterized by the appearance of a recombination nodule. So here what happened? Recombination of the genetic material takes place. So how it happened? So it happened at a site at which the crossing over, very important question always comes from this point, right? So crossing over occur between the two non-sister chromatids, homologous chromosome forming the bivalent structure, and it also shows the tetravalent tetrad structure, right? Now the non-sister chromatids of the homologous chromosome shows the crossing over, right? It shows the overlap and later form the crossing over. So crossing over is the exchange of the genetic material between the two homologous chromosome. So here we can see this at this point. What happened? The crossing over is occurring. So here we can see the blue part of the first chromosome got attached to the second, right? And this purple color, this short arm of this particular chromosome got attached to the first one. So there is an exchange of the genetic material between the non-sister chromatids. Okay. So the crossing over is also an enzyme mediated process, and the enzyme involved is the recombinase and giving the structure like this, and this causes recombination of the genetic material. So synapses formed, crossing over occur, recombination is the result, and later there is a process of segregation. They get segregated. So the crossing over lead to the recombination of the genetic material to the two chromosomes. Okay, so recombination between the homologous chromosome is completed by the end of the packetine. So packetine is the particular step of the prophase 1 in which the crossing over takes place, and leaving the chromosome linked at a site of the crossing over. So we can see at one particular site the chromosome gets, before getting segregated, there is a point there. We can see that at this particular point, the crossing over has been taken place. Right? So the beginning of diplotin is recognized by the dissolution of the synaptonymal complex. So the diplotin, the last stage of this particular prophase 1, shows the dissolution of the synaptonymal complex, and tendency of the recombined homologous chromosome of the bivalents to separate from each other except at the site of the crossover. The whole chromosomes get separated, but they are still attached. At the crossover site like this. So they are still attached at the crossover site. So this is the particular part where the crossover has been taken place, right? So at this particular point, they are still attached. So this X-shaped structure is called as chiasmata. Very, very important, right? So chiasmata is the site at which we can see that X-shaped structure, and this shows that at this particular site the crossing over has been taken place. Okay, so in oocyte of some vertebrates, diplotin can last for months or year, right? So diplotin in some of the oocytes of some vertebrates, diplotin can last for months or years. They are still get attached like this only. So the final stage of the meiotic prophase 1 is the dikinesis. Dikinesis. This is marked by the terminalization of the chiasmata. Terminalization of the chiasmata, right? So here the diplotin and dikinesis. In the diplotin, it is showing the X-shaped structure of the chromosomes, but here in the dikinesis, they get separated. During this phase, the chromosomes are fully condensed and the meiotic spindles are assembled to prepare the homologous chromosome for the separation. So spindle formation takes place and it helps the homologous chromosome to get separated. So by the end of the dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. So that also disappears. Now the chromosomes are present in the cytoplasm, right? Because there is no nuclear envelope. So here we can see this is the condition of the prophase 1. No nucleus is there. No nucleus is there because the nuclear envelope got degenerated, right? So comes the metaphase 1. Next stage, the metaphase 1. The bivalent chromosome aligned on the equatorial plate. So here we can see this equatorial plate, the bivalents, they are attached at the equatorial plate, right? And the microtubules, here we can see the microtubules from opposite poles of the spindle attached to the kinetochore of the homologous chromosome. So here we can see the kinetochores. These are the kinetochores at which the spindles are attached, right? So the homologous chromosomes separate, which the sister chromatids remain associated at the centromeres. So what happened? This sister chromatids are still attached to the centromeres, right? And they are moving towards the opposite poles. They are moving toward the opposite poles. What happened at the telophase now? The nuclear membrane and the nucleolus reappear. Now they reappear. Cytokinesis follows at this end and the diet of these cells. The two cells we can see here, right? The two cells has been formed at the result of the telophase 1. So prophase 1, metaphase 1, anaphase 1 and the telophase 1. But in the prophase 1, we have seen the different sub-stages. So that is it takes longer duration of time. So although in many cases the chromosome do undergo some dispersion, they do not reach extremely extended state of the interphase nucleus. Okay, so this stage being the two meiotic division and is called the interkinesis and is generally very short-lived. So there is a gap between the meiosis 1 and meiosis 2, but that is a very short, for the very short time, right? That is the short-lived, and there is no replication of DNA during the interkinesis. And don't get confused that there will be again a DNA replication. No, there will be no DNA replication. Okay, now here all the chromosomes which are present at the paired form. So one chromosome has shifted to the one cell and another chromosome has been shifted to the another cell, right? So the interkinesis is followed by the prophase 2, a much simpler prophase than the prophase 1. We have seen that in the prophase 1, we have a different substages, like, right? But here we are not having any substages, right? So it is quite simpler one than the prophase 1. Prophase 2. Meiosis 2 is initiated immediately after the cytokinesis, usually before the chromosome have fully elongated. So in the meiosis 2, what happened? It initiated immediately after the cytokinesis. And what happened in this? It contrasts to meiosis 1. Meiosis 2 resembles to the normal mitosis. It is very simple. It is very simple, and you can see what happens in the mitosis. It's same occurring in the meiosis 2. The nuclear membrane disappears by the end of the prophase 2. So in the prophase 2, the nuclear membrane disappears, the nucleolus disappears, and the chromosome become again a very compact stage, right? So at this stage, the chromosome align at the equator, and the microtubules from the opposite poles of the spindle get attached to the kinetochores of the sister chromatid. So again, the chromosomes are in this stage get attached to the equatorial plate. So this is forming the equatorial plate, right? And the spindle fibers are attached to the kinetochores, like this. So this is one chromosome and this is another chromosome. Now what happened? Now this spindle fibers is pulling toward this way, okay? And this centromere, it's get divided at the kinetochore, right? And this chromatids get separated like this, and it move toward the opposite pole. So but at the metaphase stage, this stage, we can see that all the chromosomes are arranged at the equatorial plate, right? And the microtubules from the opposite pole of the spindle get attached to the kinetochores of the sister chromatids. Anaphase 2, here comes the anaphase 2, okay? So it began with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together. So here you can see it is holding the sister chromatid. It is moving toward the opposite poles. So this is chromatids, and this is chromosome, right? Chromosome at the equatorial plate, that is at the metaphase 1. But this chromatid, both the chromatid get detached and it moved toward the opposite poles, okay? So it began with the simultaneous splitting of the centromere of each chromosomes, which was holding the sister chromatids together, now allowing the sister chromatid to move toward the opposite poles of the cell by shortening of the microtubules. So microtubules get shortened, okay, and that are attached to the kinetochores and move towards the opposite poles. So you can see what happened at the telophase. Again, you can see this will form one nucleus and this will form another nucleus. So it will again give rise to two cells, and this cell will again form a nuclear membrane, and it give rise to, after the cytokinesis, it give rise to the two cells. So total we are having four cells. Let's see what happened at the telophase in the separate slide. Meiosis and with the telophase 2, in which the two groups of the chromosomes once again get enclosed by the nuclear envelope. So again, it forms a nuclear envelope. Cytokinesis follows, the resulting in the formation of a tetrad of a cells, and 4 haploid daughter cells are formed. Now, what is the difference between this cell and the cell which is formed in the mitosis? So mitosis is having chromosomes. Here they are having chromatids. So they are having half the chromosome number, right? Okay, so significance of meiosis. What is the significance of meiosis? Meiosis is the mechanism by which the conservation of the specific chromosome number of each species is achieved across the generation. So, how can we say that? So, male individual is diploid, female individual is diploid, right? And in the reproductive organism, gametogenesis process takes place. Gametogenesis takes place and they form gametes. And that is also form N. Egg, and that forms sperm. Right? So this occur by meiosis division or the reduction division. Now what happened? When they get united by the process of fertilization, they form 2N. They form 2N zygote. Now from this zygote, division takes place and forms the embryo like this, and it forms the embryo. So all the cells here is of 2N, that is diploid, which is showing in the individual. So only this particular stage, haploid stage, okay, there is a formation of a zygote which is diploid, right? So it showing the conservation of the specific chromosome number in each species, right? So it also increased the genetic variability in the population of organisms from one generation to the next. So genetic variability, how the genetic variability come? We have seen that crossing over has been taken place in the prophase 1 stage, right? Prophase 1 stage of the meiosis 1. So that is the recombination. A new characters comes, okay, from this male individual and the female individual. For same character, they are having a different chromosome, a different allele on the same chromosome. So there is an intermixing of the chromosome that shows the genetic variability. So this is the uniqueness of this particular type of meiosis. Okay, so variations are very important for the process of evolution. So we have seen that in the evolution, how the new characters comes, that is because of the variation. And how the variation come? Because of this genetic variability, because of the recombination process. Okay, so this is all about the meiosis. Okay, thank you very much.